Hello, welcome to another practice session for IIT JAM 2022. This is the first practice session for biochemistry related topics, so stay tuned till the end of this video. There would be 10 questions that we would be answering in 20 minutes or so stay tuned. Now, if you are preparing for IIT JAM 2022 and you are confused about where to practice questions and where to give mock test from, then let me tell you, Unacademy is bringing a free mock test this 28th November, that means coming Sunday. So if you are preparing for this particular exam, definitely opt for this mock test, and you can enroll for free using my code AP10, and you would get all the links which are provided in the description. So for biotechnology, the test would be around 9 a.m. and this is a free test. Okay, you can use my code AP10 to get a free access. Links are all in the description. Okay, so let's begin with the questions. Question number 1. So these are biochemistry related questions. So generally they ask some informative questions from the biochemistry part. So let's look at the questions. Conversion of glucose to glucose 6-phosphate in human liver is performed by hexokinase only, glucokinase only, hexokinase and glucokinase, glucose 6-phosphate dehydrogenase enzyme. So this enzyme this particular question asks for the knowledge about isozymes, especially hexokinase and glucokinase. You need to know the difference between them. Click on the I button. But the correct answer here is both glucokinase and hexokinase, which are active in liver. The following enzyme is required for hexose monophosphate shunt pathway, right? So you need to identify the enzymes. That's kind of like a very important enzyme in this pathway, and the correct answer is option D. Anyway, if you want to learn more about pentose phosphate pathway then or hexose monophosphate shunt, you can quickly click on the I button. A detailed video explanation is provided. Two conditions in which gluconeogenesis is increased are diabetes mellitus, atherosclerosis, fade conditions and thyrotoxicosis, diabetes mellitus and starvation, alcohol intake and cigarette smoking. So gluconeogenesis is a process where glucose would be produced from other sources, like breaking down of the amino acid or taking the intermediate of the TCA cycle. Question is, what are these situations when this kind of gluconeogenesis is favored? Correct answer is option C, diabetes mellitus and starvation. In diabetes mellitus, what happens is glucose is present, like you get glucose from your food, but the problem is glucose cannot be channelized inside your tissue. At the end of the day, your tissue needs the glucose. So if it doesn't get the glucose from blood, it has to get it from other sources, and that happens. Glucose is several other components like amino acids, like Krebs cycle intermediates, gets converted to glucose in the tissues. And in starved situation, when there is a need of glucose, other stuff like amino acids and intermediates of Krebs cycle would provide glucose in order to provide energy. So that is why starvation and diabetes is the correct answer. At the lowest energy level of alpha helix of a polypeptide chain is stabilized by what? This is very simple question, but they ask specific points regarding alpha helices. So do check out the video on alpha helix provided in the I button, and the correct answer here would be hydrogen bonding, which is pretty easy to understand. A lipoprotein inversely related to the increase of coronary atherosclerosis is VLDL, IDL, LDL or HDL. So we are talking about good cholesterol, that is HDL. HDL is like a kind of scavenger for cholesterol. It takes out cholesterol and it scavenges. And LDL kind of deposits cholesterol here and there. If you want to learn more about LDL, HDL, the link is provided in the I button. All the following statements about hydroxyproline are true except there is no codon for hydroxyproline. It is present in large amount in collagen. Free proline cannot be hydroxylated to hydroxyproline. Hydroxylation of proline residues is catalyzed by dioxygenase. And the correct answer here would be option D. Now all these kind of answers can be tackled by learning from my biochemistry playlist. So I would strongly suggest click on your I button, go to the biochemistry playlist and look at the videos in that playlist. All of them are highly informative, very short. It would help you to revise. Question number 7. In adipose tissue, glycerol 3-phosphate required for the synthesis of triglycerides come from hydrolysis of pre-existing triglycerides, hydrolysis of phospholipids, dihydroxyacetone phosphate formed in glycolysis, free glycerol. Correct answer is C. And dihydroxyacetone phosphate, which is formed from glycolysis, would be converted to triglyceride under fade situation. Like when you have too much of glucose, some amount of glucose would be used for generating of energy using the glycolysis process. After a while, there is no need of generation of energy. In that situation, several anabolic stuff would happen. So it would be an energy reserve for the future. And that is why dihydroxyacetone phosphate from this pathway would be eventually converted to triglycerides. All the following statement about ketone bodies are true except their synthesis is increases in diabetes mellitus. They are synthesized in the mitochondria. They can deplete alkali reserve. They can be oxidized in the liver. Now the correct option here is they can be oxidized in the liver. Liver produces ketone bodies. Liver cannot utilize ketone bodies. Ketone bodies which are produced by the liver can be utilized by muscle, brain, kidney, heart, etc. but not the liver. And if you want to learn more about it, the ketone body related video is there. The ketone body utilization video. Okay. Question number 9. The following metabolic and abnormalities occur in diabetes mellitus except increased in plasma free fatty acid, increased in pyruvate carboxylase activity, decreased lipogenesis, decreased gluconeogenesis. So the correct answer is decreased gluconeogenesis. Actually, under the circumstance of diabetes, gluconeogenesis increases. So this is an important point altogether. Question number 10. Transfer of carbamoyl moiety to carbamoyl phosphate to ornithine is catalyzed by a liver mitochondrial enzyme. So basically they wanted the rate limiting enzyme in the ornithine cycle. Anyway, a general trend in IIT JAM is basically they ask question from the rate limiting enzymes. So for each pathway, rate limiting enzymes and their regulations are really important. Here the correct answer would be ornithine transcarbamylase. So the ornithine cycle is again provided inside the biochemistry playlist. Urea cycle regulation is also provided. Check out all the videos in the biochemistry playlist. I bet it is really useful. Anyway, if you like this video, if you think this video is useful, don't forget to like, share and subscribe. Don't forget to check out the previous year question playlist which is also provided with this video. Thank you.